So when your compound, your drug is actually is inhibiting or activating based on how you are created, it will give you a signal, and that signal is basically electronically, it is recorded. That's what happens. So that's what basically methods are. Behind the scene, they are very simple methods. So cloning and all, as you saw the pieces, breaking down the DNA to small pieces, and you can put it back together to create your own sequence that you want, and that sequence then you can use. So for example, why I'm telling you this, for example, as I was telling yesterday, and now you will see the slides. I have tried to give some thoughts in my each lecture, so how I can make the connection. So yesterday I showed you one example of the non-small cell lung cancer, right? A study that showed out 10,000 lung cancer patients, and when they studied, it showed out there were 16 different biomarkers, 16 different proteins which are there, which could be the cause for that led to about 10,000 patients who have studied cancer. Think for a second. Now, if you have to test some drugs against one of those 16 proteins, which could be the cause, you have to have a method developed, right? अब कैसे पता चलेगा ये protein A है, protein B है, protein C है? So you take that protein out, you clone, you take that protein out. So these techniques. I'm trying to make connection from that was very macro level पे कि ठीक है, non-small cell lung cancer, there are 16 proteins. ठीक है, पता चल गया. अब protein को identify कैसे करें? How do you identify कि कौन सा protein इसमें है? So these techniques, or in order to, let's say for any specific, or c-Myc is one of the protein, for example. If that protein, if you're studying that protein, you have to develop a method which is specific around that specific protein of your interest. So you can create these clones. These techniques basically help you to create those clones, and then in order to develop an assay, so that if you take a drug, if you're developing a drug which is for that specific lung cancer because of c-Myc, for example, so you have to have an assay. When that assay gives you a signal, right? In order to develop an assay, you have to know what sequence is there, and now you take that sequence and create your own assay. Jisme at the end you can clear fluorescent, luminescent, for example. So when if the function is that the protein or the drug when interacts with c-Myc, so let's say if that signal in the beginning is 100, whatever that number is, and if a new drug or an entity is blocking that, it goes down to 40, which shows that it causes 60% inhibition, right? 100 होना चाहिए था, 60 का मतलब that drug can cause 60% inhibition. But how do you know what is that inhibition? You need to have an assay. An assay basically is what you clone that cell line or cells. At the end you put a tag. Now in order to clone that, you put the pieces together. First you need to know what its sequence is. अगर किसी चीज़ को बनाना पता चाहिए ना कि exactly वो है क्या, then you can in lab you can create that. So that sequencing then helps you to clone that. So fragmentation, these techniques, for example PCR and all that, helps to break down to understand what the protein is, the cause, and then this cloning then helps to create back what you want, so that you can create an assay, and then you can do the testing of thousands of compounds or whatever needs to be done, so you know that for that specific. So इसी तरह से अगर there are 16 proteins, for example, in that case, for each one you can create this, and now you have an assay for each one of those. So cloning is a technique that helps you to create. So biologists in my lab, they do frequently this. If they are working on a specific cell line, they are specific cancer type or any disease type, they look at what is now, because there is so much advance. So many of these proteins, their sequence is known and information is available. So they take that, they create their own vector, and there are companies, you send them that this is the vector, this is the sequence that we need. They create that and they send it back to you, the cell line. You use that cell line now to study your new drugs, or you can create your own. So, but the technique which is used are the things that we are just talking, to analyze, to do that. So and again, so there are many other vectors that you can actually create. inside, you can create a bacterial artificial chromosomes, you can create yeast artificial. So expression vectors, you can create these vectors, you can design these vectors. But how do you design? You first need to know what they are in order to design that. So these techniques actually help you to do. Okay, we'll stay with Hindi and Punjabi, no Korean. So in order to do that, now that's at the smaller level, but you can actually do at high throughput. You can take a large numbers, you can put, you probably know that when you have 96 well plate, 384 well plate, now there are 1536 well plate. All these, that many wells, and you can actually put your, the things of interest in each of those wells, and you can test those. So you can actually do high throughput. So basically what it does is you can take several genes to identify which gene is being interacted. So let's say, for example, you have a drug, you create a drug in the cell. Your cell are responsive. So this is again going back to see the connection. Yesterday I showed you a slide coming from left or right, phenotypic screening or target based screening, right? So if you start from phenotypic screening, which means a cell, a phenotype, a cell, you test cell whether they survive or they die when you give a drug. So there is a specific cancer cell, let's stay with lung cancer cell line. You take new 1000 compounds, you test and see some cells, because of the function of those compounds, out of 1000 compounds, maybe the 20% cell die, in some case 80% cell will die, in case 90, 40, whatever that number is. But those cells, so now you know that those compounds are effective causing the death of the cell. Okay, that's at the micro level. You now want to understand inside the cell which genes are being affected by that. Cell, as I showed you, it's like an ocean. It's a big complex composition of many genes and protein, thousands of genes and proteins, for example. Now, so when you gave a drug and drug is causing the death of that cell, now you need to understand inside the cell which genes are affected. Right? Cell is a macro level. Now you dive deeper into genes. So you can do a high throughput screening where you can take the drug and you can actually do high throughput biology, and that helps you to again to connect what genes or set of genes are being affected. Say for example, so it is possible to do high throughput biology, and again, the functional genomics experiments you can do same way for DNA sequencing, DNA microarray, transcript at the transcript level, structural genomics, metabolomics, and I'll give you a definition of all of these, in the my second lecture today. So basically these information, and based off that there is a lot of information which now bioinformatics, because many of these genes and proteins level information has been already developed, well understood, stored. So you can actually computational way, you have that sequencing and everything is available. You can use bioinformatics in order to make connection make sense, what works, what doesn't work. For example, so I have a student in my lab. She was undergraduate from University of Toronto, cell biology. She did her, she was working at Stanford in clinic. Then she did her master's from Hopkins in bioinformatics, and now she's doing PhD in my lab. So when she has to, again, I think after there were a few people and I was just trying to learn, I think totally best is to soak in as much information as possible. So she did cell biology, went to do computational bioinformatics. Now when she is doing her PhD, she's actually very resourceful to other people. So frankly, her own research, when she looks at, she immediately goes to the computational, whatever public domain data is available, and she will try to see how much in the public domain, what type of cancer, what type of disease, and how much is available information. As a matter of fact, she helps in people, those who do not know bioinformatics, by trying to look at and analyze the publicly domain data, and she is co-authors on their work. So knowledge never goes bad. Knowledge is always helpful. So now she is the go-to person in bioinformatics. But bioinformatics, again, because she can do all of these things. While she is doing her own, she's working on lung cancer. Her PhD thesis is on lung cancer and trying to understand and try to develop and test new drugs. But how you can use the known information, so you can have the sequencing information, all of that is available. You can use bioinformatics, which is highly advanced, and that can help you now automation. Things are now more advanced. So I showed you, for example, the simply PCR and restriction enzymes, breaking it down. You can do high throughput screening. You can use, and now that's why these techniques are. And I don't know, here you probably may know in the biochemistry or biology department here or somewhere around in other places, in let's say PGI or neighboring institutes, people have these kind of instruments. Very simple. They just simply sequence and they just find out what genes and proteins are working on. But that's basically what they do. That's basically what they do. They look at the genes at the protein level or the at the metabolite level or transcript level by using these techniques, which are now automated. So one can simply have that all that information gathered in order to make better understanding or provide better understanding of what might be happening inside, if the cells of interest, the genes of interest, the proteins of interest, or the tissues of interest in that specific disease. So sequencing, you can sequence the fragment DNA. You can subclone those. Again, at the protein, this could be done at the whole genome. So you can sequence the whole genome. And so I'll give an example of, for example, in my lab, as I mentioned, we design and study a lot of drugs, from natural products to known drugs, to design our own chemical space. Now the question is to understand how in any disease type. So for example, in my lab I have people working on various different types of cancers, basically their area of interest that they have. So when let's say if there is a drug that is at the cellular level is showing very good response, that shows that it actually has an effect. Now you want to understand the gene, so you can do as a very whole genome. So it will give you a whole look at the whole genome and see what genes are upregulated, downregulated, affected, pre-treatment, post-treatment. So you can gather all that information to see if you have a drug, how is that affecting. But you can do the whole genome. So which is again understanding the DNA part, understanding their fragments, understanding what is the sequence, and putting all of that information. It could be simple at a one step at a time. It could be automated. You can do hundreds and thousands together. But you gather all this information by using the simple technique which I showed you earlier. Now things are obviously very advanced. So these can be done in a much faster way. But look at, so larger, you create the vector, as I was telling you, we can create a vector in the lab. So create a large vector. You can source the clone and you can create subclones. You can assemble the subclone to create the cells of your interest, cell lines of the interest that you have. So as a chemist, we talked yesterday about you're designing a drug, and many of you are organic synthetic chemists. You're designing whatever molecules of interest. But now to test those in the biology side. So you can do all of these, the biological side of things, and you can create the cells of interest, the assays of interest, and then at the structure level, at the function level, you can basically start, as I was saying, unrolling एक layer हटा के biology की, trying to understand at the large level to smaller level. So at the genes level, transcript level, protein level, breaking down to peptide level, breaking down to smaller clones. You can understand one by one. You can unroll so that you can go back to see what is the cause of any protein or how their protein is working. For example, there are proteins they have various isoforms, and within those isoforms there are only simply um, 10 base to 14 base difference. They are like 300, 400 base. Just last 10 to 10, that makes the difference. One could be normal and healthy, the other could cause cancer. But there is a very small piece in between that is different. In order to understand, you need to be able to chop entirely to see what is different, how they are, they are modified or changed because of the disease or in the disease. So how do we study that? So these are the techniques which are used to study that, so that it can help in now in many cases there are proteins, because of whatever reason they are modified, they are changed. So you need to first understand what change happened, how that change happened, and where that change happened, which leads to. So for example, there are 2000 around or so transcripts. 4 to 6, they work at the cluster of 4 to 6 transcription factors. How do we understand where the change is, what that change is, and what change actually is important, what change causes the cell function to go haywire and they start multiplying much faster than the normal cell and becomes cancer? At what stage it becomes cancer? Because it doesn't automatically becomes, there what leads to. So stress, for example, has been one of the environmental factor which is cause. You see tobacco. There are so many ads to see the avoid tobacco. For whatever the regulation level is, कि वो मूवीज़ के अंदर इतना स्मोक करते हुए हीरो को जिसको भी दिखाएंगे नीचे एक छोटा सा लिख देंगे कि टोबैको इज़ बैड, तो क्यों दिखा रहे हो? बट दे शो दैट क्योंकि वो अच्छा लगता है. दैट्स ऑन द साइड. बट हाउ डज टोबैको अफेक्ट योर बॉडी? राइट, कुछ तो हुआ ना. बॉडी वॉज नॉट मेड टू के स्मोक एंड चू टोमेटो. सो टोबैको से कुछ तो हुआ. सो वो क्या हुआ? व्हाट चेंज दैट हैपन? Right? So in order to do that, where that change happened, what that change happened. So if the stress level cell ke upar, see what are these? These are quote unquote environmental insults, radiation, jo chemical weapon. Now for example, yesterday I gave you an example of the first known the chemotherapy, which started from observation that the mustard gas, when mustard gas, very small molecule, went inside, created blister. कुछ तो उसने change किया ना biology पे. So what that change happened, right? So to be able to understand, as you unroll this more and more, you have to go down to the roots. Cause क्या है उसका? But how do we go there? So these are basically techniques that are used to be able to better understand the biology, and based of that to be able to then do the designing of the assays, designing of the models, designing of the techniques that can help you to better test your drugs. In case of chemists trying to design new drugs, design new molecules, design new entities, then be able to test those and without having false negative or false positive. Okay, any question? I don't know what the program is. Is there I think there's a break, right? Half an hour break, and then we meet at 12. 11:45? Okay. So, if you don't have any question, let's take a break and then come back and then I'll go over the omics part. What are the techniques, other techniques which are used to understand the proteins, the genes level? Yeah. Oh. Thank you, Karim. जो सो रहे हैं उन्हें जगा रहे हैं. देखिए मैं मजाक करता हूँ इसलिए क्योंकि लाइटर बना रहे हैं. So you take a break, you come back and we just talk again. By the way, I'm just kidding. Nobody was sleeping, okay? Don't feel bad.
Thank you, sir, for that informative lecture. We have a short tea break and kindly reassemble by 11:45.